Hello, my name is Katya Mashi, and today I'll be presenting relating bowel and psychiatric disorders using the microbiota gut-brain axis. This presentation will be based on the study Antibiotic-Induced Microbiota Perturbation Causes Endocannabinoidome Changes, Hippocampal Neuroglial Reorganization, and Depression in Mice. This research has serious implications on the world because as of 2022, worldwide, there are 10 million people that suffer from inflammatory bowel disorder. And as of 2019, 970 million people worldwide suffer from mental disorders, and understanding a connection between these could have serious implications on how they can be treated. Specifically, using mental disorders can be far easier when they're connected to a physical cause, such as bowel disorders. The main question that is asked in this study by the researchers is, when an original mouse's gut microbiome is perturbed completely, will the mouse's behavior, biochemistry, or electrophysiological properties change? The researchers think that gut microbiome has an effect on the mouse's psyche. Specifically, they believe that perturbing it causes endocannabinoidome changes. The endocannabinoidome are lipid receptors that are biologically and functionally similar to cannabinoid type 1 and 2 receptors. Cannabinoid receptors are involved in metabolic regulation, cravings, pain, anxiety, bone growth, and immune dysfunction. The experiment that the researchers proposed for this was that they introduced antibiotics to the mouse's gut and then introduced probiotics to the mouse's gut, and then they hypothesized that it will perturb the mouse's gut microbiome completely and cause subsequent psyche changes. Prior to this study's findings in 2017, the field developed a few more important discoveries over about a decade. Ivan Pavlov won the Nobel Prize in 1904 for making one of the first discoveries of the gut-brain axis. He discovered that in digestion, gastric and pancreatic secretions will release in response to sensory signals from the brain, like sight or smell of food. As of 2008, researchers discovered that the hypothalamus controls visual cues of food and how the brain responds to intake of food, and how diabetes changes the response to glucose intake. Food intake was discovered to be related to what gut peptides are present, and they are also involved in regulation of insulin secretion and sensitivity. Overall, they discovered that the gut-brain axis is involved in many processes, such as food intake and satisfaction, regulating glucose, fat metabolism, insulin secretion, and bone metabolism. As of 2015, researchers began raising mice in germ-free environments and using antibiotics and probiotics to change the mice's microbiomes. After they were raised in these germ-free environments, it showed how the microbiome development affected behavior, stress, how pain is received, and biochemical processes involving neurotransmitters. These studies also perturbed the mice's microbiome using antibiotics and probiotics, but had yet to use fecal transplantation. Also, they discovered that endocrine and neurocrine pathways play a role in changing microbial composition and behavior to affect signaling. As of these discoveries, they have yet to implement any clinical trials to determine if the findings were transferable to humans and treating human mental disorders. Something that I personally found interesting is that while I was searching on PubMed, a scientific literature database, most studies on the gut-brain axis were released around 2020. Also, the amount of studies began to increase around 2010. As stated previously, 10 million people suffer from inflammatory bowel disorder, and 970 million people suffer from mental disorders. Also, those who suffer from either of these ailments will have lower average life expectancies than individuals without them. Overall, inflammatory bowel disorders like ulcerative colitis and Crohn's disease are very deadly. Inflammatory bowel disorders causes 25 to 50% of deaths in Crohn's disease and 18% of deaths in ulcerative colitis. Along with this, around 8 million deaths occur yearly due to mental disorders. Providing treatment for inflammatory bowel diseases and mental disorders is important, and it can save millions of lives. In the study, the researchers' hypothesis is that if they subject mice to antibiotics, then use Lactobacillus casei as a probiotic, then there will be subsequent changes in the gut and brain. Now, do they have any evidence to support their question? The researchers believe that a reason there is a relation between the gut and the brain in regards to mental disorders is that the endocannabinoid system becomes dysregulated by the gut microbiome composition, and it plays a role in depression by affecting the hippocampus. Why are the researchers testing this? There have been evidence that probiotics can reduce stress, depression, and anxiety. Also, there have been reports that children show a link between gut microbiome perturbation and risk of developing psychiatric disorders in their adulthood. Based on this, the researchers suggest that the use of probiotics can be a preventative for developing psychiatric disorders. The researchers want to specifically study the cellular, neurophysiological, and the biochemical mechanisms to explain why gut microbiomes and neuropsychiatric disorders are related. The following is the methodology that the researchers used in this study. The researchers perturbed the gut microbiomes of mice using antibiotic slurry, then they used Lactobacillus casei as a probiotic. Then, the mice underwent behavioral, biochemical, and electrophysiological tests to see whether the perturbation led to changes in inflammation, depression, social behavior, cognition, neural firing, microglial activation, or alteration to the endocannabinoidome. They started with drug administration. To create the slurry of antibiotics, they mixed drinking water with ampicillin, streptomycin, and clonidamycin. Then, the mice drank the slurry for 14 days. They used a control group to drink regular water for 14 days as well. The researchers then administered the probiotic Lactobacillus casei orally with saline for 7 days. They also alternatively injected the probiotic into a separate group of mice for 7 days. After this, they began testing the mice's behavior. The mice underwent tail suspension tests where they were suspended by their tails and observed based on how long they remained immobile. The more immobile a mouse was, the more depression symptoms it showed. Next, they underwent a forced swimming test where for 6 minutes they were forced to swim and try to escape the water. Immobility was determined at minutes 4 through 6, and immobility was a sign of depression. Motor impairment was measured by a road-to-road test, where mice's equilibrium before falling onto the magnet was determined. A muscle strength test was also recorded, where the mice hung from wires and their fall times were measured. The mice's memory was tested in object memory testing. They allowed the mice to roam freely in a setting where there were 2 identical objects. Then, the time each mouse spent exploring the object was recorded as less remembrance of it. The mice's working memory was also tested using a maze with spontaneous alterations. The pain tolerance of the mice was tested using hot plates as well, and their pain tolerance was based off of their reactions to the hot plates. Their sociability was tested based on free roaming with other mice and observers noticing if they grouped with any mice or were isolated. Then, they sacrificed the mice and performed Western blots on their brain tissue. They specifically used their cortex and hippocampus. The researchers also used their fecal samples to extract their DNA and perform 16S sequencing. Specifically, they used Illumina Index and sequencing adapters. Next, they took blood from the mice groups as well as serum, and then performed ELISAs on the serum. They determined their electrophysiology by anesthetizing them and drilling holes into their skulls to record their brain activities, as well as collecting neurons from them. They also observed them under a microscope, the neurons. They measured their N-acetyl serotonin and N-acetyl ethylamine levels and their endocannabinoid levels, and then they extracted their lipids. Overall, the researchers discovered that microbial perturbation does affect multiple cellular and biochemical mechanisms. The mice that had consumed the antibiotic slurry showed an increased depression symptoms in all of the behavioral studies compared to the control mice. After administering the probiotics, the test mice had a decreased amount of depression symptoms. Also, they discovered that the microbial perturbation did not have an effect on strength or motor function, only their psyche. Test mice that did not have any probiotic treatment regained their gut microbiomes and returned to normal behavior in 2 weeks. Also, they discovered the time spent engaging in social behavior was not influenced by the perturbation of the microbes. However, they noticed that the test mice were unique in their socialness compared to the control mice. With more probiotic treatment, there was more social interaction. Cognitive performance was not affected by the test at all. Neurotrophic factor regulation was decreased in the test mice, and their tryptomyosin receptor kinase B was increased. There was a decrease in bacterial diversity in the test mice, showing an increase in proteobacteria and acinobacteria, and a decrease in Bacteroidetes and Firmicutes. An increase in Lactospiraceae was correlated with immobility time. This is one figure that I thought was interesting, which shows the bacterial diversity within the different mice. Most of the bacteria is shown within the first mouse, and specifically within the third and fourth mouse, Enterobacteriaceae are more prevalent. However, in the fifth mouse and the first mouse, Firmicutes lactospiraceae is the most prevalent. Personally, I took from this article that eating well and sustaining a healthy gut microbiome likely assists with mental health. One strength of this study is that they discovered a new area of the field. But one limitation is that they have yet to do clinical trials. The findings from the study can be used in many fields and save millions of lives. The researchers successfully proved that there is a correlation between the gut microbiome and neuropsychiatric disorders. Overall, the findings from this can be used to save many lives. Next, I prepared 5 questions for discussion. Question 1. What technological advances do you think occurred in 2020 to cause a rise in gut-brain axis studies? Question 2. What are some applications of the study that you can think of outside of treatment for inflammatory bowel disorder and mental disorders? Question 3. What would have happened if instead of using probiotics after antibiotics, they used fecal transplantation from other mice? Question 4. What are some examples of gut microbiome-related neuropsychiatric disorders that I have not mentioned? Question 5. Do you think that in humans, using probiotics as an antidepressant will work in most cases? If not, explain why you think it might not work. Here are my references that I used for this presentation. Thank you so much for listening, and I hope you enjoy the rest of your discussion.